Hi, welcome to the ZP Developer Zone. So we do the ZP Developer Zone once a week, and though I've got the date up as the 18th of August, this will be uploaded this evening, which is the 17th of August. But that said, let me go forward with this webinar. So in this webinar, we like to answer technical questions that have come in during the week. So questions this week, somebody's very interested in glucose and uric acid monitoring, I believe in the public health domain. So they've sent me a paper. I've had a look through the paper and I'll suggest some ZP technologies on that. Somebody else was very interested in a microneedle wristwatch. So in fact, on one of this, I've got a microneedle system here. I'll answer their questions as I go through, and having the watch here will actually help me think through some of the answers, or maybe illustrate some of the answers as we go through on that. Somebody else is also very interested in hydroperoxide. I think sometimes it's probably worth telling ZP about the application rather than saying, do you have this? I think it's sometimes better to say, how do I achieve this? And that probably goes the same for number one as well. It's rather than have you got this or can you do this? It's kind of like, tell us what you want to achieve and we could probably be more useful. And then just at the end, I'm going to talk about IR drop or IR compensation. This is a phenomenon where you've got a non-conducting solution, and it can lead to the distortion of voltammetry, specifically cyclovoltammetry. And I'll talk about why I think it's not such a problem when you've got form factors like this screen printed electrode. We don't suffer from it very much, but I have seen it in people's data, and I'll talk about where I think it's coming from. All right, with that said, we do have some upcoming webinars. So if you're interested in glucose and uric acid monitoring, then please come to this webinar on the 22nd of August. If I say anything and a link then appears, we do 2 in the morning, by the way. So if you're in China, come to the one in the morning because that will work best with you. If you're in the USA, come to one in the afternoon because that will work best for you. Any links, I'll make sure I post them underneath the video as I go along. There's also a wearables webinar, 28th of August. Again, depending where you are in the world, I would come to the webinar that best suits your particular time zone. And then if you're in academia and you're particularly interested in the translation, and at ZP we are particularly interested in the translation of research, and I'll make some comments about that translation today as well during this webinar. The 25th of August is the right webinar for you. When I talk about translation these days, I'm specifically using the Sensit or potentiostat. It's a real revolution in the way potentiostats actually operate, and I'll be referring to this instrument a few times in the webinar today. I generally think this is an instrument that's kind of unique in the world of biosensing and electroanalytical sciences at the moment. It's probably the most well thought out mechanically and technology-wise. It's app-enabled, it's cloud-enabled, it takes a screen-printed electrodes. It's a real platform for anyone who's wanting to do point-of-care type diagnostics. Though that said, let me go forward, and that will be certainly something we'll cover on the 21st of August. Now, but this webinar is probably going to be put online on the 17th of August, so it's only 4 days away for that webinar. And just out of interest, if you're watching this much later on, these links will probably take you to the webinar that's most upcoming, let's say. So we keep on using the same link, but we do these kind of webinars on a monthly cycle. With that said, let me go forward a little bit. So Sensit all, somebody's very interested in doing a glucose and uric acid determination. I can't quite remember the matrix that they're interested in. I mean, when I say matrix, it wasn't sweat, I don't believe, but sweat, urine, saliva, blood. These are the kind of matrices that at ZP we get involved with. Now, they were asked a very specific question. They sent me the paper. I appreciate that. I have read the paper. I have understood it. The paper allows you to detect glucose and uric acid. But it's kind of, there are lots of ways of detecting glucose and uric acid, and the paper is kind of, it's interesting, and, it shows some nice concepts, and I have definitely gone through it. I've only using these images from that paper just to kind of illustrate that we have gone through it, we have analysed it. And I think this is a sort of typical kind of paper where you actually end up, I'll define what I mean by technology readiness level number 3 in a minute, where you end up with the raw signal and you end up with a calibration curve. But that's it. That's where it ends. And then they kind of, and it's not criticism of the paper. This is just, a comment on academic papers a lot. Then you have this kind of concept of an app on a smartphone. But of course, making that app on a smartphone, you've got to do it. And I say app on a smartphone, I realize you can't see it because in fact I'm in the way of the illustration. Actually doing it and then making it robust is two different things. So you've got to first of all do it and then you've got to make it robust. So, sometimes when you read papers, I think people read them and take them at face value, but, this app doesn't exist at the moment. And, the app development is fairly straightforward. You've got to be a little bit careful with the idea that you're going to be able to just generate that app. So now what I'm going to talk about is technology readiness levels. So that said, there's something called technology readiness levels. I'm using this to comment on the paper, or not comment on the paper, comment on the papers in general. So you have an idea, that's kind of technology readiness level number 1. You start working on it, so you come up to technology readiness level number 2. So that's a basic technology readiness. Technology readiness level number 3 means you went in the lab and you did it. That is where most papers actually sort of finish at. Some of them may do a little bit further and maybe do real samples, so they may be starting to argue that they're at technology readiness level number 4. Realistically, to get things out of the lab, you need to be coming up, to 5, 6, and 7, doing technology demonstrators. So most scientific papers, so this person wants to do glucose and uric acid. They found a specific paper to do it. The specific paper does lab work. They produce the raw signal. They have a calibration curve. They have a concept for a web-based app. Actually, ironically, I can just scan glucose on this particular. That was the wrong one. Sorry, I'm just trying to scan glucose here. Let me just. I got these QR codes in front of me, and there we are. And I'll maybe I should talk about that a little bit in a little bit deeper. But I've got something in front of me called the Sense It All. It's a potentiostat, but it's more than a potentiostat. It's also a way of delivering point of care diagnostics earlier to applications than would otherwise be done. But Sense It All is at a higher technology readiness level. So sometimes it's not, the paper's useful, but actually it's going to be such an effort to raise that particular paper to a higher technology readiness level. It's actually going to be, too much. If you really want to do some public health studies earlier on, you're probably best off using a technology that is already, let's say, more robust. The ultimate goal is technology readiness level number 9, but technology readiness level number 9 could mean something like, that you've actually done enough clinical trials that you're kind of able to meet the FDA standards, let's say, in the USA. So no criticism of the paper. The paper's a fine paper, but you finish, the paper kind of finishes at TRL 3, and then to lift something like that to to TRL 7, which is where I may be suggesting that you need to get to, is actually quite a lot of effort, and actually not just effort but money, time and money. So somebody could spend sort of 5 years making something robust. Well, 5 years is a lot of time, and that time was actually money. They know in that 5 years, people were making salaries were being paid. Even if that money was coming from a kind of government source, there's still a lot of investment there. The investor just happened to be the government. So if somebody came to me and said, I want to make a glucose and uric acid sensor and I want to do it in public health quicker, I would say the paper's interesting, the paper's indicative that this could be done by the methods otherwise laid out in the paper, but to really do it quickly and to do it at a reasonable price, it's too much of a expensive, time-consuming lift to do it for the paper. That seems like a 2 to 5 year effort. In fact, if you are interested in that, we've got a Sense It All webinar, which is one of the webinars I was linking to at the beginning. Come to that and I'll talk about that that commercialization route a lot more in that paper. With that said, we are able to do glucose and uric acid. We have this Sense It All platform. In fact, I might just change screens just for a second, which I will do here. So I'm going to use this potentiostat to actually talk about cyclic voltammetry in a bit. So just that FYI, I've just put the app now into cyclic voltammetry mode. This potentiostat is in fact, I need to connect it. There we are, connect it now. There we are, it's connected now. This is now in Bluetooth connection with this, and that means I can do cyclic voltammetry on this. This will become a little bit more useful and apparent in a short while. Yeah, in peanut spectroscopy, because I'm actually going to talk about impedance spectroscopy in a short while as well. But this particular person is interested in glucose and uric acid. In fact, I scanned the wrong QR code there. There we are. Right, so now actually I can run a glucose assay on this potentiostat. So this is a very unusual kind of potentiostat because it allows both the allows you to both do fundamental electrochemical research and actually to start becoming app. When I say app, I mean analyte specific, sort of glucose, chili, ginger, potassium, pH, nitrate, etc. With that said, I'll go back to my slides just quick and say, so we can't raise that paper to technology readiness level number 7, for example, very quickly. It's going to take a lot of research, a lot of investment. You know, even if you produce a prototype, its robustness, its resilience won't be that great initially unless you really invest in it. The quickest we can do is actually just using the technologies that we already have. And if you see a link pop up, the link will be underneath the video when I finished. Question number 2 is about microneedles. The person asking the questions is super familiar with our microneedle technology. So I'll just take this off now. This is our microneedle technology. So they're super interested in this. I'll have a look at their questions now, and that will. Does the microneedle based wristwatch need calibration? Right. So first of all, we will only deliver our microneedle technology into well-funded programs. In the first instance, I believe that it will require a glucose calibration. So, for example, the way now, in fact, what I have in front of me here is I'm holding up to this camera here. This, and I know that you'll all be familiar with this. I've used this so many times. This is the Abbott Freestyle Libre 2. When you see people walking around with these, patches on their arms, often actually they are using the Abbott Freestyle Libre 2 or Abbott Freestyle Libre 3. I just saw Abbott saying the other day this has been the sort of, I don't know if it's the fastest selling something like that medical device in history. One of the problems it's got is actually the size of this applicator. This applicator is actually quite big, so there's quite a lot of plastic in putting this device on the arm, but Abbott know what they're doing, so I'm sure that they've worked out the economics on this. So the question is, let's say I had a wristwatch like this, and I was, it's microneedle, this one is, so it goes through the skin. It doesn't require an applicator. You can just kind of pull it through the skin like that. The question is will it need calibration? Now, the quick answer is, if you look at Dexcom or Abbott, when these products first came out, they absolutely required a finger stick test during the first 24 hours. Only later on did they become calibration free. And it's worth saying that in order for Dexcom to get onto the market with their first generation product, I think they spent something like $500 million US, and they were still requiring calibration. So the quick answer to your question is, yes, I think it will require calibration. How will that happen? Well, this is how it happened for everyone else's technology. You take a glucose strip, you take a drop of blood. Is that glucose strip on? Yeah. You take a drop of blood. Oh, I'm going to love this. Ow. All right, there's my drop of blood. Good thing about electrochemistry is it can be quick, so this will be about 5 seconds. And literally, this is how Abbott and Dexcom first did it. So my blood glucose is at 111 milligrams per DL. I know it's a slightly funny unit. Here it goes, 111 milligrams per DL. What you then do is actually program this to say, at this moment, my blood glucose is 111 milligrams per DL. So you can understand that's a single point calibration. And then you do that a few hours later, and then it would basically start using those single point calibrations to essentially calibrate this. Because what you end up doing is calibrating between the signal on the watch and what your blood glucose really is. And if you do that a few times, then you can essentially end up with sort of doing let's say an in vivo calibration. it sounds crazy, but that's actually how, when Abbott first went to market with something like this, that's exactly what they were doing. They would get finger pricks tests during the first 24 hours. We do have human data on this. We will only release that human data to well-funded clients. So the quick answer is we do have what's called first-in-man data. Now, you've got a certain question here about materials, shape, etc. So what I'm going to do is I'm going to tell you that there are patents. I'm going to give you a link to the patents, but I don't really want to spend this webinar sort of digging into this, to be honest with you, because there's something like 50 patents. So if it's, I'll show you where it is in the public domain, and if you can find it in those patents, then you're welcome to it. The reason I'm bringing this up is because this watch, for example, actually has 50 patents behind it. There was a company called PK Vitality. ZP was always their electrochemical biosensor developer. They went into liquidation. When they went to liquidation, ZP bought all the IP. So I'll give you a link to that. How to calculate the following. Oh, I wish I meant to, I may be able to do it. Quick answer is I've got an online calculator that shows you how to go from conductivity in millisiemens to sodium chloride. I'll put that link underneath this video. I haven't looked into how to calibrate hydration from potassium. I'm sure I can look into it, but it's going to take me some time, and then I have to check myself. So the quick answer is I know people are using potassium as a way of determining what the hydration level is, but I have not looked into the calculations, and the same for pH and ATP from oxygen. I have not looked into the calculations. I can do it, but it would take me some time, and I'm not at this moment really got enough bandwidth to do those calculations, let's say. But I will put this link underneath the, sorry, I've just realized I've just done all that talking and I wasn't actually sharing my screen. So I'll just go back slightly and just say, yes, the wristwatch will need calibration. How do you do it? By that blood glucose reading. We do have reliability data, but we will only release it to well-funded clients. Shape and tip, I'm going to give you a link to some patents. How to calculate sodium chloride. This link will take you to something I have on the website. I haven't done the calculation on how to look at hydration from potassium, and I haven't done the calculation on how to do pH and ATP from oxygen. I can do it. It would take me some time, and at this moment don't have the bandwidth. So here's that link. Here's some of the material you can find in the patents, and I will link to this particular patent as well. Question number 3 is really quite similar to question number 1. In question number 1, somebody's interested in a point of care diagnostic for glucose and uric acid. They asked, can you raise this a paper like this to the right technology readiness level? And the answer is you can, but it's really going to take a long time. By the time you've done that, somebody's already finished their PhD or their postdoc, and to raise most papers from TRL3 to TRL7 is at least $1 million, at least $1 million. Question number 3 is they just want a hydroperoxide test. What we have in the public domain will tell you that we have a good resolution of signal from 0.5 micromolar to 2 micromolar to 4 micromolar. So it's best sometimes to tell us what you're trying to achieve and in what's the clinical range that you're interested in, because then we can be, let's say, more informative. But we do have a hydroperoxide sensor. It does have good resolution in the range, 0.5 to 4 micromolar. And if you tell us more about your application and your clinical range, then we can probably be more informative. But we do actually have A, the raw data on the website, and B, you can buy this as a kit as well. But the more you tell us, the more we can be informative. This last one is, I get a lot of inquiries about screen printed electrodes. I like screen printed electrodes. We do manufacture them, and I think we manufacture them, and we do quite a good job of screen printed electrodes. But I do get sort of sent in some distorted data, and people think that it's sometimes IR drop. So I'm going to talk about IR drop, and I'm going to talk about why I think screen printed electrode data can be sometimes quite distorted. So these are ZP screen printed electrodes. This is the 501. This is the hypervalue carbon electrode. If you, and I have the instrument in front of me, if you take one of these instruments and you put 50 microliter drop on that, and you choose cyclic voltammetry, make sure I choose cyclic voltammetry. we do work with quite a lot of semiconductor companies actually who are very interested in getting into this kind of technology space. So we have worked with, for example, Analog Devices. It's on the public website. In fact, I talked to them years ago about electrochemistry and how it was the future, and they did get quite heavily into this a few years ago. With that said, you can do things like cyclovoltammetry. Now, when you're doing cyclovoltammetry on a potential start like this, you hope to get nice looking data like this. But sometimes it can get distorted. You know, the peaks actually start to separate a bit. And people will sometimes say, and you know, I can say it looks sort of uglier like this. So this sort of the classic, but if you've got some IR drop, then things can get a bit distorted. The root causes of this are often a poor placement of reference electrodes versus the working electrode. So it's good to have the reference electrode quite close to the working electrode. That actually becomes less of a problem when you've got things like screen printed electrodes like this. That's actually less of a problem because the reference electrode is actually quite close to the working electrode. So I don't think it's such a problem with screen printed electrode. The other reason for it is poor conductivity of solution. A lot of electroanalytical work, especially in the point of care space, the solutions are actually quite conductive. I mean, blood has a natural sodium chloride concentration of 135 millimolar. It's water-based. This is quite a conductive material. So we actually don't suffer from IR drop or distortion of cyclovoltammetry in my particular domain of, let's say, electroanalytical chemistry. When you've got larger electrodes, bigger distances, higher currents, then yes, you could probably have a bit more of a problem. This kind of the whole conversation about IR drop often is told from the perspective of I've got this disc electrodes. It can be gold, platinum, carbon. I'm in a solution. I have what's called a lugging capillary, a horrible term, but it's a glass object in which you have a reference electrode and you're trying to bring the tip of this capillary as close to the working electrode as possible. And if you do that quite well, then you'll have a nice cyclovoltammetry. If you poorly place that lugging, it's a horrible term, that lugging, but if you're not as close as you could be and you've got a bit of a distance here and that solution is not very conductive, then you can end up with a more distorted cyclovoltammetry. Now, as I say, in my world of screen printed electrode, we don't have a problem, at least with that distance problem, because in fact, you know, this is all quite compact. All the distances are within sort of a few millimeters of each other, and our solutions, you know, I just did that glucose reading a little while back. That, you know, my blood is conductive enough actually not to worry too much about IR drop. So I don't find it such a problem with screen printed electrodes, but people do contact us about IR drop and screen printed electrodes, and I'll first of all talk about IR drop, and then I'll talk about what I think is really going on here. So when we have a reference electrode and we have a working electrode, and we want to hold or apply a potential between that working and reference, let's say I want to have 650 millivolts. If my gap is too great, which I don't think is a problem with screen printed electrodes, or my solution is not conductive enough, which could be a problem if you're using organic solvents, then I'm just making I don't want to say a mock-up, I'm making a simulation of what's happening between the working and reference in a lot of electroanalytical experiments. You've got what's called the Randall circuit. I appreciate that, so that's a bit something to just drop that into the middle of this webinar. But the Randall circuit, you've got the charge transfer resistance. So if I was to talk about the oxidation of ferrocyanide, the charge transfer resistance is how easy is it to get electrons from that ferrocyanide to the electrode and form ferricyanide. The double layer capacitance is every time I change a voltage, when I'm doing cyclovoltammetry, I keep changing the voltage, and the ions rearrange, and the ease with which that rearrangement takes place dictates my double layer capacitance. Then I have my solution resistance. I call it RU here, and I realize RU means sort of uncompensated. Most of the time, that solution resistance in most electroanalytical work that's done on screen printed electrodes is actually that solution resistance is quite low. But I will do some calculations to see what the significance of solution resistance can be. But let's say that resistance was quite high. I might want to apply 650 millivolts between my working and reference, but in fact there's a bit of a voltage drop because my solution resistance is quite high. I'm just making this up, like 200 millivolts is actually dropped because of the solution, and then I want 650 millivolts actually across my between my reference and my working. In order to achieve that in this scenario, I actually need to apply 850 millivolts because I'm compensating for my IR drop. As I say, in my little world of electroanalytical chemistries, particularly when I'm talking about screen printed electrode, I don't think we suffer from this problem too much because the distances between the working and reference are just a few millimeters, and most of these solutions are actually quite conductive. But if you do have that problem, then you'll go from something that's quite classic to something that's a bit more distorted. So if you've got this, then you don't have an IR problem. If you've got something like this, then you possibly do have an IR drop problem. So just to do some back of the envelope calculations, you can use impedance spectroscopy to actually determine what that solution resistance actually is. So let me just say, to do the signal voltammetry, I can use a super bias. This is a potentiostat that Zimmer and Peacock produce, and actually I could do an impedance spectroscopy measurement. This will also do EIS as well. So I can do EIS in order to help me determine what my solution resistance is, and then I could sort of figure out what kind of impact that's really having on my signal voltammetry. And I'll do some calculations for you now. So let's say I did some EIS. So this is a simulation that I actually ran. It's a simulation, it's not actually an experiment. But if you're not familiar with electrochemical impedance spectroscopy, maybe I'll try and link to the video that we have online about this. And I do get some nice reviews on that video that people say it's actually quite easy to follow. But here I've done a simulation of this is a simulation of a circuit that defines the working electrode, and if my resistance here is, let's say, 100 ohms, I could have actually measured that, because in fact where this is the Nyquist plot, and where this crosses the x-axis here, that is 100 ohms there. Now let's say I had a solution resistance that was 10x that, and actually 1,000 ohms. No issue. This is the same simulation now, but see that that's now saying 1,000 ohms. So I can go from 100 ohms to 1,000 ohms, and I can I can measure what my solution resistance is just by doing an EIS experiment. This is a simulation, by the way, that I would kind of, if I was doing an oxidation of ferrocyanide, this is what I would expect the Nyquist plot to kind of approximately look like. So what I'm saying here is I can get a sense of how much I'm suffering from IR drop by actually just measuring the solution resistance. Now if I do a back of the envelope calculation that voltage is equal to current times resistance, a lot of the times when I'm using screen printed electrodes like this, the most current we're really going to be getting is microamps of current. Now microamps of current is 10 to the minus 6 amps of current, and I'm talking here, let's say my solution resistance is 1,000 ohms, 1 kiloohm. It's quite a lot actually, but 10 to minus 6 times 10 to the 3, that actually gives you a 10 to minus 3. So my voltage loss is 1 millivolt, 10 to minus 3 volts. So what I'm saying very quickly to you is you have to have quite some solution resistance in order to really suffer from IR drop on a screen printed electrode. And now I think our ZP screen printed electrodes actually give quite nice side voltammetry. We've put a lot of effort in over the years, especially when they're used in solutions that are quite conductive. And we do, like many other people who are making screen printed electrodes, we do benefit from the short distances between the reference and the working. I do on occasions get sent images like this, and I'm showing very distorted data. And actually, it's not the IR drop that's a problem with these kind of, this is not a ZP screen printed electrode. It's the conductivity of the electrodes themselves that people are using. I've helped somebody in the past. They were doing paper-based screen printed electrodes, and they really suffered from poor conductivity of their electrodes. So it's not IR drop that's the problem. It's actually high resistance along the conductive track. So it's almost like you're using cables that have got high resistance in them. So people see the distortion, they think, oh, it's my screen printed electrode, I must have an IR drop problem. No, you've got a resistance in your tracks on your screen printed electrode that's the problem. It's not the solution itself. So let me just do a quick summary. To lift a paper that's really at technology readiness level number 3 to something that's higher is a lot of investment and a lot of time, and it will take probably more than 3 years most of the time. And most of the time people's PhDs or postdocs are over. So you have to jump on a platform that's already quite mature. I'm obviously biased, but you know, we've got the SenseIt platform that's both can be used for analytes and it can be used for fundamental electrochemistry. If you want to dig into that a bit deeper, we've got a few webinars in the next couple of weeks. Look at the links underneath. Microneedle wristwatch. You had some questions about calibration. I talked about the glucose meter, for example. You had some questions about the needles sizes. I've given you links to patents, specifically one of the patents where they actually do call that out. To insert, maybe I didn't make that point, you know, the microneedles are actually here. We are looking for semiconductor companies, by the way, who are willing to become involved in this quite heavily. And then to insert it, you just pull on the strap and that will then insert. Question number 3 was hydrogen peroxide. Tell us more about the application, but otherwise, if you're not wanting to tell us about the application, you can buy the technology online. And then I think people think that they're having an IR drop problem with their homemade screen printed electrodes or with their electrodes that they've sourced from a third party, but it's actually the conductive tracks of the ink that are used to make the electrodes is actually the problem. If you've got any questions of ZP, don't hesitate to reach out to us. Otherwise, take care. Okay, thanks very much.